All right, part two of our test review will include a review of non-Mendelian inheritance. What we'll see here in non-Mendelian inheritance is that not all of genetics falls into a very nice, neat package where you have a simple dominant and a simple recessive gene that go to war against each other to figure out which one is going to be expressed. Sometimes there are some special situations to where maybe those phenotypes blend together, or maybe there's multiple dominant alleles that are all expressed at the same time, or what happens if you end up with a trait that's on a sex chromosome. And so that's where all of these special cases come in, and we call all of these things non-Mendelian inheritance or non-Mendelian genetics. And so we talked about 4 different examples of that. And the first example is called incomplete dominance. And in incomplete dominance, there is really no dominant or recessive alleles at all. Instead, if you end up with a homozygous individual, obviously they're going to show one trait, but if you end up with a heterozygous individual, then they end up showing a blend of phenotypes. And so kind of the gold standard example of this is the snapdragon plant. And the snapdragon plant has 2 main phenotypes, that being CW, which codes for white, and then CR, which codes for red. Now, if you'll remember, I said don't pay too much attention to what this letter is. That C may stand for color. I don't really know what it stands for, but this W here, this superscript, is going to tell you what the phenotype is. And so this R here tells us what this phenotype is. Okay, so if we have a red snapdragon plant, that means that this one's genotype must be CR. So it's homozygous for the red gene. Excuse me, the red phenotype. If we have a white snapdragon plant, then it must be CW and CW. So it is homozygous for the white phenotype. Now, if we crossed these, then every single one of the offspring would end up with the genotype CR-CW. And so in incomplete dominance, this is where we end up with a blending of both of these colors. So instead of it showing the red being dominant or the white being dominant, instead we mix the red and the white, and then we end up with a third phenotype, which is pink flowers. Right? So in incomplete dominance, we are blending phenotypes. Okay. The next example of non-Mendelian inheritance that we have is called codominance. So here, instead of not really having a dominant allele, here there are multiple dominant phenotypes. And if the dominant phenotypes are together, each dominant phenotype will be expressed equally. Keep in mind there's no blending here. The multiple dominant alleles will be expressed equally. Okay? And our gold standard example here is blood types. And there's several different blood types that you can have. You can have type A, you can have type B, or you can have type O. There's even another example called type AB. Okay, and if you have type A blood, then you have 2 possible genotypes. You could have what's called IA, and A is dominant, and then you have this little recessive I here, or you could end up with IA, and that's all going to code for type A blood. And the A just refers to this antigen that's going to be found on the outside of your red blood cell. If you have type B blood, then you could have IB, recessive I, and then you could have IB, IB. And both of those are going to code for type A and type B blood. Where the codominance comes in is if you somehow manage to get this genotype IA, IB. So instead of just one of those letters being dominant over the other, so instead of A being dominant over B or vice versa, instead you're going to end up with both of those. So both of these are going to show up in equal amounts as our phenotype. And so we call this a fourth type of blood. We call this type AB blood. Okay, and then just in case you're wondering, this last type over here, type AB, the only way you can get that is by getting the 2 recessives. Okay, but once again, the idea of codominant is that there's multiple dominant alleles that both show up at the same time. So both A and B are going to be expressed equally at the same time. And that's our example of codominance. The next example that we have of non-Mendelian inheritance is called multiple allele. In multiple allele genetics, there are several different alleles that have levels of dominance. And where we see this is in rabbit fur color. And so there's 4 different phenotypes that you could have. You could have what's called a full color gene. You could have chinchilla, you could have Himalayan, or you could have albino. And what we're going to see here is that the full color gene is dominant to the chinchilla gene, which is then dominant to the Himalayan gene, which is dominant to the albino gene. Okay, so if we ended up with a rabbit that had the genotype C plus CCH, we're not going to blend these two together. This one is going to win because it is higher up on the hierarchy of dominance. So here the full color allele would win. We'd end up with this rabbit here. If we tried to cross CH and then C, if we look at our levels of dominance here, the CH would win, and so we would end up with a Himalayan rabbit. If we had a rabbit with the genotype CCH and then C, once again we'll refer to our levels of dominance and we'll figure out that CCH would be dominant to C, and so this genotype, this phenotype would win, and so we would end up with a rabbit that shows chinchilla fur coat. Right? So once again, multiple allele is different levels of dominance within the phenotypes. Okay. Now the next one is a little bit more complicated. It's called sex-linked inheritance. This is what we spent a whole day talking about. This occurs when you end up with genes that are located on the X chromosome of sex chromosomes. Now, the sex chromosomes are typically the last pair of chromosomes, and they're the ones that determines whether the organism is a boy or a girl. And so what we'll see there is that if you end up with what's called an X and a Y, you are a boy. If you end up with what's called 2 X chromosomes, then you are a girl. Now, what I want you to notice about these is that the X chromosomes are typically very large. The Y chromosome is typically very small. Now, normal inheritance and normal genetics patterns are followed if these genes are homologous here within the Y chromosome and within the A chromosome or the X chromosome. So we can look at these, we can compare those, and that's fine. Normal dominance rules follows. But what if you end up with a gene up here and then there's nothing over here on the male chromosome? Then what happens? Well, what happens is that whatever this says, that's what the boy is going to have. We're not necessarily going to run into that problem here with the female because they have a pair of homologous chromosomes and 2 X's, and so we can follow normal dominance patterns. This is typically only going to happen with males because they don't have this information here to compete with this information that's here on the X chromosome. So let's do an example to see what that's all about. Hemophilia is a typical disease that's associated with being sex-linked. The normal allele is capital H, and then the recessive allele, little h, stands for hemophilia. If you have 2 recessive alleles, if you're a girl for hemophilia, then you'll have it. If you're a male, you only have to have 1. Okay, but let's see how that works together. So we're going to cross a hemophiliac male, and remember males are XY, with a normal female, which is XX. Okay. Now, anytime that you get to a problem on a test or homework and it says sex-linked, the very first thing that I want you to do is I want you to write down XY and XX, because from there we can figure out what the genotype is going to be based on what phenotype the person has. So here we have a hemophiliac male. Now, in order to have a hemophiliac male, all he has to do is have 1 H, or 1 recessive gene on his X chromosome. There's not going to be anything out here on the Y because that Y is not big enough and it doesn't have all of those genes. Okay. Now, if we have a normal female, then that means that she's going to be homozygous for the dominant trait, which is normal. So she's going to be right here. So XX and then 2 H's. And so now we just put this into a Punnett square like we would anything else. So I'll put the female information here, X dominant, and then I'll take the male information and put it up here and call that X recessive, and then I'm going to call this Y. And now let's just fill this in like any other Punnett square. I'm going to drag the Y's down. I'm going to drag this X lowercase h down, and then I'm going to drag these X dominant H's across. Drag this one across. And so now I've filled in my Punnett square. And now I've figured out all the possible genotypes that we could have for these kids. And let's take a look and see what that means. The first thing we take a look at is the X and the Y's and figure out, okay, is this a boy or a girl? Well, the two X's means that this is a girl. Here I've got an X and Y, that means that this is a boy. Here I've got an X and an X, so that means that this is a girl. And then I've got an X and Y, that means that this is a boy. Then the next thing that we'll do is we'll take a look at what the actual alleles say. So here I have one dominant and one recessive, so the dominant trait wins, which is normal. So that means that this is a normal girl. Here I've got one dominant and then nothing over here on the Y. So whatever this is, that's what this person exhibits. So this is what we call a normal boy. Then down here at the bottom, once again, we have that dominant trait there on the X and nothing on the Y. So this is once again a normal boy. And then here on the girl's genotype, we have a dominant paired with a recessive. So she will be the dominant normal. So here, these parents have a 0% chance of having a kid with hemophilia. All four of their possible genotypes code for normal. Okay, now let's do another example. So now this is saying that let's take a normal male, and we'll call him XY. And if he's normal, then this X is going to contain an H. And then let's cross that with a hemophiliac female. So if she's going to be hemophiliac, that means that she's X lowercase h, X lowercase h. All right, now let's put that into our Punnett square. I've got X lowercase h, X lowercase h. Let me change colors, and we'll go X dominant, and then Y. And now let's fill this in. I'm going to drag my Y down, drag my X H down, and then now I'm going to take red and let's fill this in across. So I've got X recessive, and now I've got X recessive and X recessive. Okay, so now let's analyze. Here I've got two X's, so that means that this is a girl. I've got an X and a Y, so this is a boy. This is XX, so this is a girl. And then this is XY, so this is a boy. And now let's look here. I've got one recessive and one dominant. So the dominant wins. That means that this is a normal girl. Here I've only got one allele, which is the recessive for hemophilia, and then nothing over here on the Y. So that means that this boy is now a hemophiliac. He has the disease of hemophilia. This boy down here is going to be the same story because he has a recessive trait here, nothing on the Y. So this boy now has hemophilia. Okay, and then last but not least, this girl over here, she has one recessive and one dominant. Well, the dominant wins, and so this girl is what we call normal. All right, hope that helps you study for your test and do well on your homework. Thanks for listening.